So next I have the huge pleasure of introducing someone from my own laboratory. Dr. Nathalie Feeley is an early career scientist who undertook her PhD in one of the UK's foremost colorectal cancer laboratories with Professor Ian Tomlinson. I'm delighted to say she got tempted into cholangiocarcinoma research, and she's been looking at the use of AI and AI tools. One of the really important things, obviously, in the CCA community is generating tools that we can share between us to accelerate the momentum of research. And hopefully she's going to dispel some of the myths and suspicions some people have about using AI and how we can leverage this for our basic science research.
Firstly, just a huge thank you to Helen specifically and for the whole AMMF team for inviting me. This is such an inspirational room of people, and I am so honoured to be here and to be speaking to you today. So, first and foremost, I am primarily a wet lab scientist with a sprinkle of bioinformatics in there, and I work for Rachel. She's a consultant surgeon and principal investigator. So it's really our aim is not to spend too much time doing really repetitive analysis. So this project is an example of how AI can be implemented into a research project to automate it, to make it more efficient, and so we can spend more time doing more useful things in the lab and for Rachel clinically. So hopefully this presentation it is an example of how this can be done, and it will just enable maybe other lab groups to consider how they can use or implement artificial intelligence in what they do to further automate what they do and to make it more efficient, and then just encourage collaborations between wet lab scientists and bioinformaticians and data scientists and mathematicians so you can find out what is possible and how AI can be used. So this is an investigation of how we can automate the annotation of biliary populations in H&Es, or otherwise referred to as ductular reactions by pathologists. So ductular reactions are really interesting to us as a lab group because ductular reactions are really prominent in ductular obstruction. They result in reduction or stoppage of bile flow and are characteristic of multiple diseases, including those that predispose to cholangiocarcinoma, including PCS. So ductular reactions are particularly interesting because they're a correlate of disease severity, so the percentage of ductular reaction across the liver can be used to establish how severe the disease is. We can visualize them quite easily, and they are also quantifiable. They're characterized by a proliferation of biliary epithelial cells, the presence of hepatic progenitor cells, fibrogenesis, and the infiltration of inflammatory cells. And importantly, ductular reactions can be modeled in mice using something called a DDC diet and genetic editing, so we can mimic the effects of human bile duct obstruction in mice. And the way that we visualize ductular reactions or biliary populations in mouse livers was described really nicely by Tim on the first day. We take a really thin slice of the liver and we mount it onto a glass slide, and then we do something called H&E staining. So you can then scan in your glass slide using something called a nanozoomer to create it into an image so you can see it and visualize it on the computer. And then we upload it into a software called QPath. And in QPath, you can really zoom into these H&Es and you can have a look at these biliary populations and you can assess the morphology of them. So this is a manually annotated biliary population that is done in QPath. Now, the problem that we have with manual annotations is that this is the size of a H&E. This is the size of a mouse liver. This image is absolutely huge. So I'm not even sure you can see it very well on this image, but the image that I showed you on the previous slide, it's actually highlighted in the red box in this H&E. So you can only imagine how time-consuming it is to annotate and analyze an entire H&E, and then you have to times that and multiply that by the number of mice that you're interested in using. So it's a time-consuming process. This is really subject to variable accuracy. I might annotate something differently to the way Rachel annotates it. There's human error involved in this as well. And I've got to say, it's not very mentally stimulating. So in other words, it's a very boring thing to do. And we'd much rather be doing other things with our time. So the solution, I was able to set up a collaboration with someone called Kai. He's a computational applied mathematician who I'd witnessed using computer vision before. So his background, he did a master's in computational applied mathematics, and he utilised computer vision to visualise and to select text in old historic maps of Edinburgh. So what he managed to do was get the computer to visualise, to select the text. He managed to remove the text from the maps, and then even replace and repair the background of the images underneath. So I asked whether this would be possible for us to do, and he thankfully said yes. So what is computer vision? So computer vision is a field of artificial intelligence that allows computers to interpret and analyse visual data. So it has different roles. You can use it for different things. Firstly, image classification. So it can assign a label to an image. Is this a cat or is this a dog? Secondly, it's very good at object detection. So you can see, right, it will detect where that dog is in that image. And then thirdly, it can perform image segmentation. So it can decide pixel by pixel whether that pixel belongs to a dog or whether it belongs to a sofa. And this is the bit that we're really interested in. So image segmentation is, does this pixel belong to a biliary population or does it not belong to a biliary population? So one method of carrying out computer vision is by using a convolutional neural network. And I don't want to skate, this is just maths. It's basically, the first thing that it's really important to understand is that an image is made up of pixels, as I just said. And every pixel is represented by something called an RGB number. So an RGB number is just a ratio of red to green to blue. So this is the way that a computer sees an image. So this is really, really zoomed in. But pixels are just represented by numbers, and this is how the computer sees an image. It's not like how we see an image. And a convolutional neural network will apply filters to an image. So a filter is just a grid of numbers, and this grid of numbers will scan across the image from left to right, or however you design it, and it will do maths, and it will detect very simple features. And this is an example of the grids that are responsible for vertical edge detection and horizontal edge detection. And this is the structure of a convolutional neural network. So the very early layers will involve very simple filters. Like I just said, they'll scan for very simple features. But later layers might combine these really simple features and simple shapes to identify more complex shapes and more complex features. So for example, a convolutional neural network might be designed to recognize faces. Early layers within the convolutional neural network will scan across and look for very simple features and be able to recognize and identify very simple patterns, followed by more complex shapes, followed by facial recognition. So the first thing that we wanted to do in our project is look for already existing convolutional neural network architecture that is specifically designed for medical image segmentation. And this is where we came across DuckNet. So DuckNet is a convolutional neural network architecture that was published in 2023. It looked more up-to-date than its predecessors. It looked like it performed very well. It's very good for in-depth feature selection. It uses residual downsampling, so less computational power, and there isn't any external data required. So some convolutional neural networks, to get them to work, you have to train them on hundreds of images of the thing that you're interested in detecting, whereas this, we could use our own data to train this model to recognize biliary populations. So the next step in this process was to create, probably this is the most painful part of the entire process, is to create our training data. So training data needs to be absolutely perfect because we don't want this model to be making any mistakes. So our first task was to annotate every single biliary population in that H&E that I just showed you. So this involved creating over 500 annotations within this training data of H&Es, but hopefully will pay off in the end when we get it to automate the rest. So now we have our training data looking as best as we can, we're going to show it to the model. We divided the image into 75% training data and 25% validation data. And the validation data is used to test how well the model is generalizing to biliary populations that it's never seen before. And you can see in the figure on the right-hand side that the model progressively improves per pass of the data through the model. And a pass of the data through the model is also called an epoch. So we can measure how well it's training by using loss scores, which is just a measure of how well, of model error and how well the model is performing. So it can be seen in the graph that the training loss largely decreases as the training progresses and as the model trains on more and more samples of the same image. The validation loss, however, plateaus after about epoch 50, so about 50 passes through the data, the validation data stops improving. And after this, it's instead sort of just fitting, the training data is sort of overfitting to itself. So the model has seen the answers before, and it can perform very, very well because it knows what the answers are. It doesn't know where the biliary populations are in the validation data. So this, after epoch 50, this is where we have an optimized computer vision model that can be taken forwards and applied to further unseen H&Es. So we wanted to test this model, so we produced the model, we optimize it, and now we want to test it. So the way that we wanted to test it is by taking 10 H&Es and taking samples, so squares of 10 different H&Es. And what we did was we very nicely asked someone called Dan Field, who is a clinical pathologist, to annotate these 10 samples, and we wanted to compare our model's annotations to Dan's gold standard annotations. And this is one of the results. So on the left-hand side, you can see Dan's annotations, our clinical pathologist, and on the right-hand side, you can see our computer vision model's annotations. So you can see visually that it is performing really quite well. These are some more examples of that, and on the right-hand side, again in the yellow, is our computer vision annotations. So, okay, we can see that visually this is working quite well, but we want to quantify it just to be absolutely sure. So we use something called a dice score. So a dice score is calculated by doing 2 times the overlap of the area of Dan's annotations and our annotations divided by the sum of the area of Dan's annotations and our annotations. And with this, you get a number. And our number was mostly always between 0.8 and 0.9. And this number is particularly special because it says that this is performing very good segmentation that is widely accepted for clinical applications. So our model, it works really quite well. So future directions for this, we would really like to just do some more checks on this model. So what we would like to do is give the mice again a DDC diet and see and visualize this increase in biliary populations over time, and then let the mice recover from that and see a decrease again, which is what we would expect. And also correlate this with other markers of liver injury, so something like keratin 19, where this again should increase over time and then decrease, and just make sure that our automatic annotations are correlating with something like keratin 19, which you would expect to see performing that way. And then very importantly, the most important thing that we want to do with this is apply it to our work. So in our lab, we study IDH1 mutations and IDH2 mutations and their function in cholangiocarcinoma evolution. And looking at Dutch reactions is something that we do all the time. And hopefully we'll be able to apply this technique and just save us that time, make our research much more efficient so we can speed it up, and hopefully at some point that will benefit patients. So the key message is I really hope that this helps you to start thinking about how your research can be made more efficient and automated with AI, and just encourage those collaborations and discussions to find out with mathematicians, with data scientists, to find out what is possible, and hopefully that will lead us to helping patients in the future. So I just want to say thank you to all of the people that I work with, particularly Rachel, and for Kai for making this possible, for Dan for all his annotations, and Tim for listening to my talk the other day. So thank you very much.